Hello students, welcome to another video session. In this session, we are going to solve some of the questions which you found difficult in your AIET 4 examination. So let us start with our first question. So here it is. Consider the following regarding the reasons for the fact that the cyanobacteria are kept in Monera and not in plantae. Okay, so one second. So our options are, they are prokaryotes. The cell wall of the cyanobacteria has peptidoglycan. They can fix atmospheric nitrogen. Okay, so these are the three statements. And the correct explanation would be 1 and 2 only, 1 and 3 only, 2 and 3 only, 1, 2 and 3. All right. So let us take some of the aspects that defines the characteristic features of prokaryotes, and then we will talk about the cyanobacteria. So when we talk about prokaryotes have a very unique characteristics in with respect to their cell wall composition. So their cell wall composition is made up of peptidoglycan. When we compare it to the plant kingdom, it is made up of cellulose, right? So there is the first difference. And the second difference is the presence of your membrane bond organelles. So the membrane bond organelles are absent in your prokaryotes and present in your eukaryotes, right? And there are several characteristics of the prokaryotes that are very peculiar, like the packing of the DNA, right? And their cellular processes is quite very primitive compared to that of eukaryotes. Cyanobacteria, when it comes to this debate of whether to put cyanobacteria in prokaryote or in a plant kingdom, it was because the cyanobacteria undergoes the photosynthesis like higher plants, and it has the same chlorophyll pigmentation. So that's what makes us look cyanobacteria as more of a plant, right? So but it is very primitive organisms, and it has primitive cell organelles. So that is why it was put in prokaryotes. So the reason why they put cyanobacteria is because they are prokaryotes, and the cell wall of the cyanobacteria is made up of peptidoglycan. And the ability to fix atmospheric nitrogen is not the subject here, because it will not justify whether we have to put it in prokaryote or eukaryotes. Even several eukaryotes does the same perform the same function. So that is why we eliminate the third option as a criteria for putting cyanobacteria into Monera, not planting. So the correct explanation would be 1 and 2 only. Okay, so this is our right option. So let's go for the next question. In Pinus, the stem is branched. Roots have fungal association in the form of mycorrhiza. The male and the female strobili are born on different trees. Of the above statements, only one is correct, only two is correct, only 1 and 2 are correct, 1, 2, and 3 are correct. All right, let's talk about Pinus. So Pinus is a gymnosperm, we know that, and the characteristic features of the Pinus is very well given here in our options, that the stem is branched. So if you look at a Pinus tree, you can see that their stem is quite branched. It is very visible to us. And the roots system of the gymnosperms have a very special association with the fungal feature or fungi, which we call it as mycorrhiza. So what they do is, it's a symbiotic association where the fungi helps in deriving lots of nutrition, as well as it gives a lot of antibiotics and awareness of the environment to the pinus tree. Okay, so this beautiful symbiotic association appears in many of the gymnosperms, and this is called as mycorrhizal association. Okay, and the female and the male strobili are born on the same trees. Are born on the different trees. It's even actually it is on the same tree. So our third option is wrong. So our first and our second option is correct. So 1 and 2 are correct. Our option 3 is correct for this particular question. The pyrenoids are made up of. Our options are proteinaceous center and starchy sheath, core of protein surrounded by fatty sheath, core of starch surrounded by sheath of protein, core of nucleic acid surrounded by protein sheath. Okay. So let us talk about pyrenoids. Pyrenoids are basically found in chloroplasts. So pyrenoids, what they do, how they appear is, if you see the pyrenoid, they basically appear as a proteinaceous core with the starchy granules. So pyrenoids appear like this. So there is a uniform matrix, homogeneous matrix. Among a homogeneous matrix, you can assume that there are granules of starch or starch granules that are spread across, and then at the center there is proteinaceous core or proteinaceous center. So those are pyrenoids. So our option for this, right option for this is proteinaceous center and starchy sheath. So coming to the function of pyrenoids, pyrenoids actually function in a mechanism called as carbon concentration mechanism. Okay, carbon concentration mechanism, or CCM we call it as. So it helps the chloroplast to get abundant amount of chlorophyll, abundant amount of carbon dioxide to perform, the photosynthesis. Okay, so that is our first option is right. Pyrenoids are proteinaceous center on a starchy sheath. So, which of the following is not the floral characteristics of family Fabaceae? And our options are calyx, are not the floral characteristics of family Fabaceae. Calyx, sepals 5, gamosepalous, imbricate estivation. Corolla, petals 5, polysepalous, vexillary estivation. Andrecium 10, diadelphus, anther, dithecus. Gynoecium, ovary is inferior, bicarpellary, unilocular with many ovules. Okay. So when we talk about the characteristics of Fabaceae, Fabaceae is like the beans, right? Leguminous plants are represented by your Fabaceae. So basically there are three families, and these three families have the similar characteristics where you have five petals, they're gamma petals, they're implicate estivation, and then you have your placentation, that is axial placentation. Ovary is superior. Okay, ovary is quite superior. And then you have 10 andrecium, which is diadelphus, anther is dithecus, it goes into vexillary estivation. Okay, so these are the characteristics of, our Fabaceae family. Here the gynoecium is given as ovary is inferior. So ovary is not inferior, ovary is superior, and it is divided into many locules. Okay, it is multilocular. So that is why our fourth option is wrong with respect to this question. Okay, so let's go for the next question. Bulliform cells are found in the dorsiventral leaves and help in photosynthesis, are found in dorsiventral leaves and help in minimizing water loss, are found in isobilateral leaves and help in maintaining water loss, are found in isobilateral leaves and help in photosynthesis. Sure. So when we talk about bulliform cells, what are bulliform cells? Bulliform cells are found mainly in monocotyledonous cells. So they are kind of an epithelial cells with a very large vacuole in the center. So the role of bulliform cells is to prevent water loss. So what happens is whenever the favorable conditions are there, bulliform cells absorb a lot of water and they help in stretching of the leaves. So they get this, like a grass or any of these leaves to get exposed very well to the sunlight. Okay. And whenever there is very harsh weather and there is too much of sunlight, so what it does is it loses the water, thereby helping it to curve the leaves. Okay, the leaves get curled inside, avoiding the loss of water through precipitation. Okay, so that is the role of bulliform cells. They're transparent, they're epithelial, no, okay, and they are found in the upper surface of your monocotyledonous plants. Okay, so these are the functions of bulliform cells and where they look like, or what they look like and what they perform. Understood. So, according to given options, the upper surface of the leaf is called as isobilateral surface. Okay. So this, see, if you take the monocot leaf like a grass, okay. So at the, you can see that there is a central rib, right, or central vein. So across the central vein, these bulliform cells are quite accumulated. Okay, so that is where they are present. So our option is are found in isobilateral leaves and helps in maintaining water loss is the right option for bulliform cells. Remember this, it's a very important thing. So they are also cylindrical cells, they are cylindrical looking cells which are found in monocots. Next one. Which of the number of following statements amongst the following regarding the phloem tissue of plants are correct? Okay. Gymnosperms lack albuminous cells and sieve tubes. The companion cells are specialized parenchymatous cells. Phloem parenchyma is absent in most of the monocotyledons. Okay. Phloem fibers are generally absent in primary phloem. Okay, so when we talk about the characteristic features of your phloem, okay, so here it is. So the gymnosperms lack albuminous cells and sieve tubes. Actually, this is a wrong statement. They do have albuminous cells and they have sieve tubes. Okay, that is our first option is wrong. Next one, the companion cells are specialized parenchymatous cells. So this is absolutely right. Companion cells are modified kind of parenchymatous tissue which performs the conduction of the sieve plants in dicotyledonous plant, right? Then phloem parenchyma is absent in most of the monocotyledons. See, we do, this is absolutely right. We don't have phloem parenchyma in most of the monocotyledonous plant. It is found in dicot actually. Phloem fibers are generally absent in the primary phloem. This is also right. So 1, 2, 3, 2, 3, 4 are right with respect to this. So three statements are right with respect to this question. So that's why we choose option 3. Identify the incorrect statements. Cork cambium is peridome. Ground meristem is a cortex and pith of a dicot stem. Procambium is a primary vascular tissue. Protoderm is a vascular tissue of a monocot stem. Okay. So let us talk about identifying the incorrect statement. First of all, we have to talk about primary meristem. Okay. So when we talk about primary meristem, okay, we have three different types of primary meristem. One of the meristem is called as protoderm. Okay. Then we have procambium. Okay, and then we have peridome. Peridome, procambium, and then we have is the protoderm. Sorry, protoderm. Yeah, okay. So what happens is each of these meristematic tissue derivatives. I mean, like when the cells are born, they differentiate into different specialized tissues, right? For example, if you take about peridome, peridome differentiates into epithelial. Protoderm differentiates into parenchyma, sclerenchyma, and then you have your collenchyma. Whereas you have your procambium, procambium will give rise to the primary ground tissue, that is your xylem and phloem. Okay, so these are the origin of the primary meristems. Like primary meristems, that is where they come across. So looking at the statement, that is cork cambium is peridome. Okay, ground meristem is a cortex and the pith of a dicot stem. Then another one is procambium is a primary vascular tissue. Then again, protoderm is a vascular tissue of a monocot stem. So cork cambium is a peridome. Cork cambium is not a peridome. Okay. So that is why our option 1 is wrong, whereas the rest of the option is right. Okay. Next one. Crops such as tomatoes and bell pepper are allowed to grow in carbon dioxide rich environment. They show higher yields because our options are they show an increased rate in photosynthesis at higher carbon dioxide concentrations. They can respond to high carbon dioxide conditions even in low light conditions. They show C4 pathway for carbon fixation at high carbon dioxide in the limiting is the limiting factor in such plants. The only carbon dioxide is a limiting factor in such plants. So coming to this question, when we talk about plants like tomato or bell pepper, the reason why they are grown in greenhouses where there is a lot of carbon dioxide and there is proper temperature and proper environments and less pathogens is because the more the carbon dioxide is there, the more is the photosynthesis. The more is the photosynthesis, the stronger is the root and the stem, and the stronger is the and more is the yield. So that is why our right option is they show an increased rate in photosynthesis at higher rate of carbon dioxide concentrations is the right option. Understood? So consider the following where the photosynthesis is going on a steady phase. A typical experiment with the light on and the carbon dioxide is combined with ribulose bisphosphate REBP, okay, to produce 3-phosphoglycerate 3-PG. Then suddenly the source of the carbon dioxide is eliminated. What changes occurs in 3-PGA, 3-PG, that is 3-phosphoglycerate, and REBP would occur. Whenever we cut down the concentration level of the carbon dioxide during this photosynthesis reaction, what happens is the first molecule that gets fixed the carbon dioxide gets fixed into the system of a C3 plants is through 3-phosphoglycerate, that is 3 carbon containing compound. So that is why that process gets eliminated when we reduce the concentrations of carbon dioxide. So the concentration of 3-PGA falls eventually. Okay. Then what happens to the RUBP? The RUBP levels will quite raise. Okay, with the resultant of the decrease in carbon dioxide. So that is why our option 2 is correct. Okay, let's go for the next question. Compared to retinal, chlorophyll can be described as a pigment that has a narrow absorption range but high efficiency, narrow absorption rate but low efficiency, wide absorption range but high efficiency, wide absorption range but low efficiency. So when we look at this question, why do we consider chlorophyll as a photosynthetic pigment? Because it actually taps a very particular frequency of or wavelength of our light, okay, for the process of photosynthesis. Here, what it does is it absorbs this energy in a very high efficiency. We know that 680 and 700 nanometer of light that is absorbed by our chlorophyll pigments. Whereas retinol do not do that. It has broad spectrum, but it does not have the efficiency. Chlorophyll has a very narrow efficiency, whereas high narrow absorption range, but very high efficiency. That is why it's a very ideal photosynthetic pigment. Okay. A non-membrane bond organelles found exclusively in animal cell is. So we have spherosomes, glycoxysomes, and then we have peroxisomes, and then we have centrioles. A non-membrane bond organelle found exclusively in animal cells. So in animal cells, a non-membrane bond organelle that is exclusively found is peroxisomes. So peroxisomes, they contain the vesicular-like structures which contain these hydrolytic enzymes which have the capacity to digest certain things. Okay, so they help in storage of these digestive enzymes. So it is found only in the animal cells. So our answer is peroxisomes. Spherosomes, glycoxysomes are two of the different storage bodies which are found in even both plant cells and animals. Centrioles are found in only animal cells, whereas the role of the centrioles is quite different, and it is not an organelle. Okay, it's an assembly of microtubules. So that is why we don't consider it. Okay, our option 3 is right. Which of the following does not help in excretion in Periplatina america? So fat bodies, uricoglucose, uricose glands, and then we have nephrocytes, none of the above. So when we talk about the excretion mechanism in cockroach or Periplatina america, the most of the job of eliminating this toxic waste comes from Malpighian tubules. Apart from Malpighian tubules, what we have is the fat bodies, uricose glands, and nephrocytes. Okay, nephrocytes are found in Malpighian tubules. Okay, so they help in their ciliated epithelial cells, which help in filtration of the blood impurities. So all three of them are involved in excretion, whereas that is why none of the above. So which of the does not help in excretion? So all of them help in excretion. So none of the above. Okay, is the correct option. So bipolar neuron cells in human beings is found in brain, spinal cord, embryonic stages, on olfactory epithelium. So what is bipolar? See, when you talk about neurons, you have unipolar, bipolar, and multipolar neurons based on its location and based on its function. Okay. So, for example, if you take unipolar neurons are basically present in your brain and the spinal cord, and multipolar neurons, like bipolar neurons are found in some of the receptions, such as retina, such as the olfactory epithelium of your nose, and gustatory epithelial cells, where there is detection of sensory organs, right? So these are neurons. So when you talk about bipolar neurons have two cytoplasmic extensions. Okay, so that will help in conduction of neurons in two directions. So that is called bipolar neuron, and it is mostly found in olfactory epithelium. Most of the sensory systems, like even retina is an example for a bipolar neuron. Okay, presence of bipolar neuron. Next one. Chronic autoimmune disorder affecting neuromuscular junction, leading to fatigue, weakening, and paralysis of skeletal muscles is called as. myasthenia gravis, gout, arthritis, muscular dystrophy. Okay, so when they're asking about a chronic autoimmune disorder that affects the muscular junction, that is neuromuscular junction, leading to this paralysis. So basically, one of the autoimmune diseases which we can come across is myasthenia gravis. In myasthenia gravis, what happens is a particular antibody is produced. Understand this. See, usually what happens if this is the new nerve junction and if this is the muscle junction, usually when the nerves have to tell the muscles to do a particular job, they have to send a message. And that happens through a neurotransmitter. So when these neurotransmitters are released by the uh, nerves, they go and attach to a specific receptor that is present on your muscle. So these receptors are completely destroyed by the antibodies, which are produced by this autoimmune, oh, excuse me, which are produced by this autoimmune, uh, wait, the antibodies are produced to destroy this reception. When the reception is destroyed, usually acetylcholine cannot be produced, and that thereby that destroys the entire communication between the nerves and the muscles. That leads to paralysis of the skeletal muscles, where your neurons do not have the control over it. Okay, so this is the answer. Whereas the gout is not all related to with respect to neuromuscular junction disorder. Arthritis is the degeneration. It is all even arthritis is also a autoimmune disease, but here the degradation of bone cells will take place. And then you have your muscular dystrophy. In muscular dystrophy, what happens is it's a loss of muscles because of a genetic disorder. So that is why muscular dystrophy is not an option. Option one is correct. According to the sliding filament theory, what happens? Actin and myosin filaments slide over each other to increase the length of the sarcomere. Okay, length of A band does not change. I band increases in length. The actin filaments slide away from the A band, resulting in the shortening of sarcomere. Very relevant options are first one and fourth one. When you look at this, according to sliding theory, sliding filament theory, what happens? Actin and myosin filaments slide over each other. That is absolutely right. Now, increase in length of the sarcomere. The sarcomere contracts actually. Okay, that leads to contraction of the sarcomere. So that is why the last option, the actin and the filament slide away from the A band, resulting in the shortening of the sarcomere. Yes, the sarcomere actually kind of contracts, resulting in it shortening. So that is what leads to the muscle contraction. Okay, but then this is our right option. I band actually increases in length, and the A band does not change. A band actually, it shortens. So that is why our second option is wrong. And even the first option, it leads to the increase in length of sarcomere. It's actually a decrease in length of sarcomere. So our option four is right. Next one. Consider the following characteristics: air bladder, operculum, and viviparity. Then we have the characteristics present in bony fish include 1, 2, and 3, 1 and 3 only, 1 and 2 only, 2 and 3 only. So for this question, we know that our bony fishes, they have air bladder. That is why it can relax in water, and it has the buoyancy to maintain its body. It has operculum, the covering of the gill slits, and then it is viviparity as well as oviparity. There are many characteristics of bony fishes, the types of scales that are present, and then the type of scales also vary, and the type of the behavior in the fresh water also changes, the migration pattern changes. Anyways, all three characteristics actually are the characteristics of bony fishes. But if you're confused about oviparity or viviparity, they exhibit both viviparity and oviparity. Most of them are oviparous and some of them are viviparous. So 1, 2, and 3 are the very right answers for this question. Okay, next one. What causes the onset of puberty in males? Increase in secretion of testosterone by testis, decrease in secretion of testosterone by testis, increase in secretion of GnRH by hypothalamus, decrease in secretion of GnRH by hypothalamus. So when we look at this question, we know that when we talk about puberty in males, immediately we'll point out to the increase in testosterone levels that leads to the secondary sexual characteristics development. But if you trace back to the hormone mechanism of how the testosterone is produced, it all begins with the activation of your pituitary glands, right? So for that, a GnRH, that is gonadotrophic releasing hormone, has to be produced that stimulates the anterior pituitary to produce the luteinizing hormone and FSH, and thereby it goes and stimulates your Leydig cells as well as your Sertoli cells, one for testosterone production, one for spermatogenesis. So all of these chain reactions are beginning at GnRH. That is production of GnRH, that is gonadotrophic releasing hormone, which is produced by hypothalamus. So that is why our option is 3 and not the increase in testosterone level by testis. Even though it's very relevant option, but most of the puberty characteristics are mixed characteristics, which is more responsibilities are there apart from just production of testosterone. So that is why our third option is correct. The partial pressure of oxygen is equal in. It's a very interesting question. Atmospheric air and alveoli, alveoli and oxygenated blood, alveoli and deoxygenated blood, deoxygenated blood and tissue. So the partial pressure of oxygen is equal in where? Where do you find it equal? So the right option is basically it's deoxygenated blood and tissues. When you talk about the deoxygenated blood that is present in your bloodstream, and if you observe the tissues, they should have the same partial pressure of oxygen. That is why there is no movement of gases that is happening between the deoxygenated blood and the tissues. So here, since it is equal, there is no loss of oxygen. Only the carbon dioxide is removed. The partial pressure of carbon dioxide is different in the deoxygenated blood as well as the tissues. Apart from that, the oxygenated blood and the oxygen partial pressure in the deoxygenated blood and the tissues remain same. That is why it is able to carry from tissues to your lungs for the oxygenation. Apart from the from the given options, this is the right option. Okay, so that is why our option 4 is right. Contraction of diaphragm leads to increase in the volume of thoracic chamber in the anterior posterior axis, increase the volume of the thoracic chamber in the dorsiventral axis, and then decreases the volume of thoracic chamber in the anterior-posterior axis, and then you have increased the volume of thoracic chamber in the dorsoventral axis. So we know that when it comes to mechanism of the respiration, three things are involved. One is the diaphragm, other one is the internal intercostal muscles. We have external intercostal muscles and internal intercostal muscles. So we have internal intercostal muscles that are playing a very important role. So when you talk about contraction of the diaphragm, when the diaphragm contracts, usually during the respiration process, usually see diaphragm contracts and it moves downward. When it moves downward, the chest cavity lifts upward and then it expands forward, right? So this leads to the increase in the volume in the thoracic chamber, and that expansion happens in anterior-posterior axis. So that is why our first option is right. Okay, next one. The immune responses in the older people are weak due to the degeneration of thyroid, pineal gland, adrenal gland, and thymus. So when we talk about the immune mechanism, weakening of immune mechanism, one of the most important immune organs we have was thymus. So thymus, which is present just below the breastbone and above the heart. So what it does is in the beginning, when the birth, you have a very large thymus and it decreases by the time you come to the puberty. Okay, and it disappears gradually after puberty. So now the thymus is a very, very important place for the development of T lymphocyte cells. So since older people has completely disappeared thymus, so the T lymphocytes or the cell mediated immunity decreases gradually, thereby leading them to contract many diseases. So the option is thymus. Okay, so right option is thymus. Next one. A hormone that is an antagonist to the parathyroid hormone is secreted from thyroid, adrenal cortex, hypothalamus, and parathyroid itself. So one of the functions of your parathyroid hormone is to create a lot of balance in your body, homeostasis, growth, metabolism, etc. But one of the key antagonist which suppresses its activity is calmodulin. Okay, so this calmodulin is produced by your thyroid, which is also called thyroid hormone, which acts like an antagonist or which suppresses the activity of your parathyroid hormone. So that is why our option 1 is correct. Okay, so glucocorticoids do not. So here are your options. Stimulates gluconeogenesis, causes lipolysis, causes proteolysis, stimulate cellular uptake and utilization of amino acids. So when it comes to glucocorticoids are actually the ones which help your body to manufacture energy. So for that, what they do is they break down certain substances, complex substances in the form of gluconeogenesis, which causes lipolysis, breakdown of lipids, breakdown of proteins into peptides and amino acids. But what they do not do is they do not suggest your cells to take up or utilize this. The only job is to produce energy. Okay, make sure that they manufacture energy or break down certain complex forms. Understood? So that is why glucocorticoids do not involve in cellular uptake and utilization of amino acids. It's not the function of glucocorticoids. So our option 4 is right for not the function of glucocorticoids. Okay, next one. Which of the following are non-steroid hormones but still do not act through secondary messenger system, are small non-polar and can be taken orally? Prostaglandins, estrogens and progesterone, insulin and glucagon, thyroid hormones. Okay, T3 and T4. So when it comes to non-steroid hormones, and it's a growth hormone, and it acts as a secondary and do not act as a secondary messenger system, and they're small and non-polar. Okay, so one of the most easiest options that we can pick is thyroid hormones, right? Thyroid hormones are non-steroid, okay, and they are also promoting growth, and they do not act as a secondary messenger at all, so act through a secondary messenger, so they directly go on impact. So that is why T3 and T4, you can see that the patients who are suffering from hypothyroidism or hyperthyroidism, they take it orally. Okay, so they are non-steroidal. That is why our option 4 is correct. Next one. The proximal convoluted tubules is lined by the simple cuboidal epithelium, simple columnar epithelium, simple cuboidal brush border epithelium, simple cuboidal brush border, simple columnar brush border epithelium. So when we talk about our nephrons have very unique characteristics in the amount and the different types of cells and the composition of different cells in different places. And after your Bowman's capsule comes the proximal convoluted tubule, and proximal convoluted tubule has a very specific function where it removes a lot of glucose is recovered, a lot of salts like sodium and potassium is recovered. So here what happens is for this, the movement of the filtrate has to happen very periodically, and it has to increase the surface area for the absorption, right? So that happens through cuboidal epithelium. What kind of cuboidal epithelium is? It's a brush bordered cuboid epithelium because it moves the filtrate in a very specific direction, and it also makes sure that it can pick up these small ions or glucose, okay, etc., which is present in the filtrate for the recovery. So that is why our option 3 is right. Okay, thank you very much, students, and I'll see you in the next video. Hope this video solution has helped you. If you have any suggestions, please let me know. Thank you very much.